this morning to present the Fialco Awards. There'll be one this week, and the committee ended up proposing two people for the awards this year, which is not unprecedented, but it's unusual. So we'll have one talk this week and another one next week. The Fialco Scholar Awards are, of course, in memory of Phil and Helen Fialco. Phil was a medical geneticist who worked, did a lot of nice original work on cell origins of tumors. He also was administratively and leadership active in the department, was the service chief at the VA and vice chair of the Department of Medicine early in his career, then became the department chair for about 10 years, and then became dean of the School of Medicine. Helen was always a friendly and vivacious partner, and they were a great team. They sadly were killed in an avalanche while trekking in Nepal, and this award was set up by the department in their honor. A list of previous awardees, now including the two for this year at the bottom right, dating back to the time of the award was set up in the late 90s. As shown on this slide, several of these individuals are also members of the committee who selects the awardees. That committee who selects the awardees is chaired by Conrad Lyles, who's the vice chair of the Department for Research, includes members of Mary Claire King, Doug Powell, Randy Curtis, Chris Sorowitz, Ollie Press, Tom Hahn, Rudy Rodriguez, and Henry Rosen. Picked this, the two awardees for this year. So let me move ahead to introducing our speaker for this morning, who is Aude Chapuis. She's fundamentally Swiss. Her family is from Switzerland. She, however, was born in Oakland, California, and so I quizzed her about that this morning. Her parents were postdocs at Berkeley, and they were here for a couple of years and then moved back to Switzerland when she was about 2. Her education and medical training was in Switzerland, predominantly at Lausanne, and she came back to the United States in the early 2000s to work at the Hutch, particularly in the lab of Bill Greenberg, and has been remarkably productive through that time, and much of that work she'll review. She's received many grants and honors already this early in her career, and so we're pleased to add the Fialco Award to that list of her awards. And she'll be talking on harnessing the therapeutic potential of adoptively transferred cells. And so please join me in welcoming Aude.
Thank you very much. Thank you very much. So I could have also named my talk, Can we learn from patients who progress on T-cell therapy? So that already gives you an idea that I'm not only going to talk about successes of T-cell therapy, but I'm also going to talk about what we can learn from patients who progress of the failures of our treatments. So to give a quick overview of what I'm going to talk about today, first of all, we do T-cell therapy, so I'll talk quickly about why we're using TCR-based therapies to target cancer, lessons from a cancer model, the Merkel cell carcinoma, and then I want to talk about a little bit of our experience in the real world targeting self-antigens and what we can do now from what we have learned in order to overcome the barriers of the immune system targeting cancer. So everybody knows how a CD8 T cell works. Essentially, the CD8 T cell carries a T cell receptor, which is very specific for a specific HLA that carries a specific peptide. These peptides are presented, are intracellular usually, and presented on the surface of the HLA. There's a whole bunch of HLAs. When you do match transplant, you have to match those. This is a very specific key and lock phenomenon. There's a co-lock, if you will, which is the CD8 receptor. The moment that this structure forms, the CD8 T cell can be activated, secrete perforins and cytokines, then kill the tumor. The tumor dies. That's in the ideal situation, and the the T cell goes on to do some serial killing. Now, overall, it's a little bit more complicated than that. You have the TCR-MHC interaction, but you also have a signal 2, which is a co-signal that needs to happen if the T cell, if you want to prevent the T cell to be tolerized. Usually, one example is CD28 that binds to CD80-86. It turns out that cancer cells don't have a lot of these co-signals in order to provide a signal to the T cell, probably accounting for the fact that a lot of T cells are tolerized to the cancer that is in the patient. And we'll talk a little bit about that. Now, in the types of constructs that are in the clinic, I'm sure everybody has heard of the CD19 CAR T cells for ALL and lymphomas, and the fantastic successes that these constructs have. Now, I'm going to start by describing just the CAR T cell, so you understand a little bit the difference with the T cell receptor. CAR T cell is the CAR construct is a man-made construct, which is the external portion of an antibody recognizing a surface antigen on the tumor cell linked to the intracellular portion of a T cell receptor. It also carries the signal 2 within itself, meaning that just one signal, which is the antibody binding to the surface protein, will generate 2 signals, the intracellular, so the TCR portion, and the signal 2, which is here characterized by CD28 or 4-1BB, which are classical signal 2s that the CAR T cells carry. Now, this is an endogenous T cell receptor, so it's a much more complex structure, 2 chains. This is completely natural, and it recognizes the HLA molecule presenting an epitope. These epitopes, unlike the CAR T cells, which has to recognize something on the surface of the cell, these epitopes can come from inside the cell. And the T cell can recognize this because every cell in the body has a mechanism to go fish these peptides, these pieces of proteins, of whatever the cell contains, and presents them to the surface. The characteristic here is that we do not have a signal 2. So this is really just the natural 1 signal of the T cell receptor. And I'm gonna focus on that. It turns out that a lot of the cancer antigens are actually intracellular. The majority are, and so this is why we have decided to focus on to try to ameliorate this type of treatment. I am going to talk today, so there's a bunch of antigens that you can target with the T cell receptor. I'm going to talk today about a viral antigen, a viral model antigen, an oncogene, but there's a whole bunch of overexpressed self-antigens, MART1, which is in melanoma, and I will talk a little bit about WT1, which is Wilms tumor antigen 1, which is expressed in leukemias. We have done a lot of T cell therapy. A long time ago, we started by doing what we call endogenous T cell therapy, where we would take T cells from a patient. We'd take a patient with a specific disease. We looked at melanoma, leukemia. Actually, back in the day, we also looked at HIV, taking these cells by way of the leukapheresis. And so we know that there are some cells within the patient that will recognize the tumor. So we found a way to tease those cells outside the body. And indeed, we can do this with pulsed dendritic cells. This is done within 4 to 6 weeks in a cell processing facility. We can then sort the cells that we're interested in using a tetramer, expand them, and then infuse them back into the patient. So I want to talk a little bit about what constitutes a dependable cancer immunotherapy model. And the reason is that we want to figure out, if we want to see and study how T cells work, We want to study a specific disease which has the following characteristic, which is aggressive. Obviously, not everybody's going to let us do that in patients who have good prognosis. That there's a signal response to immunotherapy, meaning that those tumors tend to respond to modulating of the immune system. We want to target an antigen that is not shared with other healthy cells to avoid off-target toxicity. And we would like also to target a tumor antigen that drives carcinogenesis, which is really the driver, and has a limited repertoire of antigens to target. And about 5 years ago, we started working in collaboration with Paul Niem and David Cole to target Merkel cell carcinoma, which I will explain why has most of these characteristics. Merkel cell is an aggressive skin cancer. It's got a higher disease-specific mortality than melanoma, and the incidence is rising pretty rapidly. We know that in 80% of cases, it's driven by a virus called the Merkel cell polyomavirus. And this virus, most of us have it on our skins, except that there's 2 mutations that must happen to the virus for the virus to cause the Merkel cell carcinoma, and it involves integration in the genome and loss of its ability to replicate itself. So these, when integrated, these T antigen, there's 2 spliceosome, with a shared common T, a unique small t, and a large T antigen. These are persistently, they are necessary for the survival of the cell and of the proliferation, that they're obligate expressors, and they are the drivers of the cell. And they have a limited amount of amino acids, so a limited span of protein that we can target. These cells are Merkels are also sensitive to the immune system. Kelly Paulson showed that these in patients who had brisk T-cell responses inside the tumor, these patients had a tendency to do much better. And about 50% of patients will actually respond to checkpoint inhibitors, suggesting that these really are sensitive to T-cell therapy. We identified a number of epitopes, so pieces of protein that we can target with our T cells with different HLA restrictions. So we now have a bank of targets that we can use for Merkel cell carcinoma. And interestingly, the virus-specific Merkel cells have very little other mutations. So really, when the tumor is driven really by the virus, and there's very little else that happens. This is not the case for virus-negative Merkel cell carcinoma, which have much more of a mutational load. One of the particularities of Merkel cell carcinoma is that it downregulates MHC class 1. Previously, I showed you that you need the HLA for the T cell to recognize the peptide HLA complex. And a lot of these virus-specific, virus-driven cancers will have a tendency to downregulate HLA. This is a reversible problem. We can use interferons. By injecting interferons in the tumor, we can upregulate MHC class 1, and low-dose radiotherapy will also do the same. We started out very simply by saying, okay, well, let's try to transfer some T cells in these patients. And we devised a plan where we would do MHC class 1 upregulation and then give 2 infusions of cells for patients with metastatic Merkel cell carcinoma. And our initial results were somewhat disappointing, where we saw most of the patients progressed pretty rapidly. Only 1 patient had a complete remission for about 13 months. We then started to look into combining our T cells with checkpoint inhibitors, with the idea that the T cells were not sufficient. So we would need something to lower this threshold of activation of the T cells to have an effect. So we combine the same regimen, and with that, we started to see really, really nice responses. Here I'm showing a patient with a rapidly growing neck node, with other lesions also. That neck node was actually radiated to upregulate MHC class 1, but within 9 months after the T cell therapy, both the radiated site and the non-radiated lesion also decreased. So overall, we saw now much better responses with 3 complete remissions ongoing and a partial remission. And in order to figure out whether our T cells had done something, we started to look at whether the first question was there persistence? I'm not showing this. Yes, there was persistence in the blood. But then the question was, well, is there persistence in the tumor? And T cells, endogenous T cells, carry every one of them carries their own T cell receptor, which we can use sort of as a barcode. At the time of T cell generation and timic selection, T cells will select a V gene, a D gene, and a J gene. They will combine. The cell will excise its DNA, so it's not present anymore. And every cell can now be identified by a unique region that can be sequenced. And indeed, this is what we started to look at. We looked at our infusion products, and here I'm showing a patient who had a complete remission, who we show that the infusion product is composed of T cell clonotypes. So these T cells all recognize the same thing, except that they are different, that they have different T cell receptors. And when we looked at what the patient had in the blood before the treatment, we saw that the red and the blue clone, the red and the blue clone were present, but at very, very low quantities. And in the pretreatment biopsy, we saw absolutely no T cell infiltrating the tumor. So this particular patient had the right inguinal node here. So this was radiated. He also had a whole bunch of other nodes around in that inguinal region. And he got T cells and the checkpoint inhibitor. And after about 6 months, he had a complete remission. We were lucky to get one biopsy of the same node 1 month after the start of treatment, and then 6 months. When we biopsied that lesion 6 months later, it was just a tumor bed. There was no actual tumor in there. So what happened to our clonotypes, and indeed when we looked at what clonotypes of our infusion products had actually reached the tumor, we saw that 9 clones from the infusion product specifically located to the tumor, much less in the blood. So it was really a specific localization. So they were infused, they got into the blood, and then they specifically went into the tumor. And 6 months later, only this black clone persisted, suggesting that this one might have been very critical for tumor control, since there was absolutely no tumor in the last biopsy. Another way to figure out whether our T cells are doing something is whether we see epitope spreading. So what is that? That means that if we infuse T cells, we have the idea that they would target the tumor and release new antigens that the immune system could then build a response towards. And indeed, this is also what we saw in this trial, where we had patients who had de novo CD4 responses. So we were targeting a CD8 with our T cells, and de novo CD4 responses and expansion of new CD8 responses, suggesting that now the immune system of the patient was now seeing other aspects of that particular tumor. So not all patients respond, and actually we found out in the course of this year, actually, so this very recent data, that we are learning a lot more from patients who progress on T cell therapy. And I'll walk you through this. So here we had a 55-year-old man who had Merkel cell carcinoma, very aggressive. He started out we gave him T cells here, I'm not showing, but this is the start of treatment of the T cells. And it didn't really, what happened was that the T cells persisted. Were very high levels of T cells. At about 40% of his CD8s were specific for the Merkel, but he still progressed on that. So we started to add anti-PD-1, and that we felt don't work clinically. So then we started to add anti-CTLA-4. Basically, he got the kitchen sink. And lo and behold, he started to have a very progressive but measurable response that lasted a whole year. And all this time, we had nice persisting T cells, which targeted a B35 epitope of Merkel. And then after 1 year, he started to progress on exactly the same treatment, and there was nothing we could do. His disease was extremely aggressive, and he ended up dying of his disease. So why? Kelly Paulson did a really, really nice job of, in my lab, did a really nice job of figuring out why this was. And here I'm just showing the persistence of the T cell. So he had a really robust population. So you would say, well, did our T cell do something? They were there, yes, but did they actually do something? Was it MHC downregulation? And we looked with a pan-HLA. We saw that there was no difference before and after in the MHC class 1 expression, so it was expressed. So the antigen must have been presented. The T antigen by antibody here and also by sequencing of the epitope that we were targeting was still present. So all the elements were present. So she also did some exome sequencing, which did not reveal any mutations. So we had absolutely no idea why this patient had progressed. At the same time, we started exploring single-cell RNA sequencing, which is now available commercially on the 10x platform. Quickly, what it does is that these gel beads have a whole bunch of primers in them, and you combine them with the T cells. And the bottom line is that you get single cell, so a transcriptome of analysis of single cell, and you can analyze about 10,000 of these cells, and this is in collaboration with Jason Bilas. So every transcript now, RNA transcript from the cell is barcoded to each cell. So every transcript in each cell has its own barcode. And then there's also a UMI, which allows you to quantitate how many transcripts for the same gene. It's not very deep sequencing, so you get an idea of what gene, but you cannot analyze the mutations of the genes. So this is really, you can use it to have a broad idea of what is going on. And so we took 4 time points of this patient, and here they're superposed, so you can see that they cluster together. So when you analyze all this data, the PCA analysis ends up in what we call a Tisney plot, which looks it's a unbiased clustering of the T cells. So all these cells will have different transcripts. And they will cluster together. And when you put all these 4 time points together, you can see that you get the same map. And this was the people helping us, Rafael Gattardo at in VID. You can now categorize these clusters and see what they are, and you can look at your favorite genes. For example, you can look at CD3, where it characterizes T cells, and you can see that, right, so they cluster around here. And K cells are up here, so less CD3. Granzyme is secreted by activated T cells and K cells. They're up here. You can look at macrophages, CD14, they're here, and B cells. And when we looked at this, here I'm showing, is it moving? Yes. Okay, so what you can see is that at the time of response in this particular patient, there was a very characterized, or some T cells, CD8 T cells, clustered together. And they didn't cluster with the rest of CD8 T cells. They just clustered together, meaning that their genetic profile was very different from the T cells. And this was only present at the time of response. And when we look at these T cells, we saw that they were highly activated with overexpressed gene suggestive activation, metabolism, and cell division. And they were very, very different from the T cells. And they were only present at the response, suggesting that something had happened in this patient to get this activated population of T cells. We correlated that with what happened at the tumor. The tumor before was cold. There was really no T cells, maybe a little bit in the periphery. At the time of response, the tumor was infiltrated by activated HLA-DR positive T cells. And again, at the time of progression, the tumor again was cold, but the T cells not entering. We looked at whether the T cells that we had infused actually reached the tumor, and indeed they had. The blue clone here, which is the dominant clone infused into this patient T cell clone, was also present at the tumor, suggesting that so we've got activation in the blood, correlation with infiltration of the tumor. So what happens at the tumor level? And here we looked at the tumor before and the tumor after. And you can see that these two tumors don't match, don't overlap at all. So the tumor at the time of progression was extremely different than the tumor at the before any treatment started. This is not the case for other microenvironment cells like tumor infiltrating lymphocytes, activated macrophage, and fibroblasts. And here I'm just showing the movie so you can really see the separation. So why was that? And indeed, when we looked at the most differentially expressed genes, we saw that the tumor before and after was really, really different. So how could that have happened? When we started looking one at the other at all these genes, we saw that HLA genes were very much overexpressed. And we started looking specifically at genes that were targeted by our B cells, by our T cells. When we looked at the HLA-A, so this is a non-targeted antigen, we look at the tumor before and the tumor after, you can see that there's not really a lot of difference. This is not the case for HLA-B35, which is the HLA that we were targeting with our T cells. So basically the tumor hereafter had completely downregulated HLA-B35. This was absolutely not detectable by IHC because we have a pan-HLA. This was extremely specific and could be reversed by putting interferon gamma or azacitidine, which has tendency to allow upregulation of HLA. We could partially reverse this. This was not at the genome, so this was not inside the gene, and it was not even in the promoter of HLA-B. So the tumor now had found a way to epigenetically escape the immune system. So by looking at the response in this tumor, now we can say, yes, our T cells certainly did something, but the tumor found a way to escape. So we were very happy to know that our T cells did something, and that certainly encouraged us and is encouraging us. There were also a number of challenges with endogenous T cell therapy. The non-gene modified T cells products rely on the patient's endogenous T cell repertoire, and this can be damaged or partially deleted depending on what tumor it is. It takes a long time to generate, and we had actually some people who had progression by the time we were generating the T cells, and suboptimal of expansion of the T cells expanded in vivo. So what we want to do really here, and we can use these road-tested or tryout tested T cell receptor to benefit other patients. Essentially, finding out which T cell receptors are the ones that are mediating the responses, isolating their T cell receptor genes, and putting them into a lentiviral vector, and then transducing cells from other patients, so somewhat conferring them a new immunity before adoptive transfer. And that's exactly what we did for one of the patients who had a response. And indeed, the benefit of isolating TCRs, they were quite potent. When we look at potency of the TCR, we look at one thing called CD8 independence. So basically that they can function just by linking the MHC without the CD8 co-receptor, and they function in CD8 and CD4 T cells. And indeed, they function in CD4 T cells with the benefit of additional cytokines, which are important for CD8 T cell help. So that looks pretty promising, and those constructs will be going into the clinic in the next year. So switching gears a little bit, I want to talk about, I talked about this is a very beautiful model, our Merkel cell carcinoma, but there are other, most antigens are self-proteins that are overexpressed in tumors. So I want to talk a little bit about the experience that we had targeting Wilms tumor antigen 1. And here we're a little bit beyond because we've actually done T cell transfers with TCR modified gene products. So a little bit of background, Wilms tumor antigen 1 is overexpressed. It's a gene overexpressed in many tumors, including AML, CML, MDS. Higher levels correlate with worse prognosis. Many AMLs express this gene, especially the high risks. There is low level expression in normal adults, although it's a gene that expresses during the embryonic development and then is shut down. But still, some mesodermal tissues, such as CD34 progenitor cells and kidney podocytes, for example. How do you define high-risk AML? So these patients have unfavorable genetic or molecular abnormalities. They're refractory and relapse, basically needing a lot of chemotherapy to be put into first remission. And they have therapy related, so the AML developing after chemotherapy, for example, for a treatment for another cancer. Their probability of survival is very poor, and that's why most of them go to transplant. So we had actually designed a trial about 5 years ago where and this is when I was a postdoc in Phil Greenberg's lab, to target WT1 in the post-transplant setting with T-cell clones at that time. So basically just one T-cell, endogenous T-cell with just, the product was stemming just really from one cell. And we had seen some effect, but really this was characterized by short persistence of the transferred cell, and also there was a wide variability in the avidity of T-cell products reflecting the endogenous repertoire of the donor. These cells were generated from the donor. So we decided to improve this, improve the TCR affinity, make it the same across the board, and also optimize the substrate cells. Tom Schmidt and Phil's lab isolated TCRC4. It's a random name. And so what it was is, so when we plugged in, so basically when we transduce donor cells with TCRC4, they had an avidity that was comparable to the higher avidities that had been administered to the patients on this first trial. They lysed fresh leukemia cells, so we were happy it was efficacy. However, it took a long time to isolate this TCR, at least more than 2 years. I think here I could probably say 3 years. This T-cell TCR is then introduced into a viral vector, and we use it to transduce matched donor cells. So essentially, patients would come for transplant. They had a matched donor identified from the G-mobilized leukapheresis, from the donor donation. We isolated EBV and CMV-specific cells for this particular trial. So why? Remember, we're going into the post-transplant setting. In the post-transplant setting, there's a lot of graft-versus-host disease. We want to try to prevent that. A lot of T-cell, the T-cells that cause graft-versus-host disease are probably in the naive repertoire. So we thought that by taking as substrate cells, so selecting the cells that we're going to plug in our second TCR as EBV or CMV, we would improve or reduce the chances of us causing graft-versus-host disease in the post-transplant setting. And also EBV especially has a big population of central memory cells, so they're younger cells, which we know have a better chance of surviving long-term after transfer. And indeed, this is what we did. So you take the cells, transduce them, expand them, sort on the double positive. So now these cells have 2 TCR, their endogenous EBV-specific TCR and the new WT1 TCR that we're putting in, and infuse that into the patient. The process took about 4 weeks, and the patients were eligible to receive this as soon as the products were ready. There were 2 arms, so there's 2 types of, so the patients who did not relapse at day 28 after their transplant, so that was they received the cells as prophylaxis to try to prevent recurrence. We know that these patients will recur with a high probability. And what we call the treatment arm, those are patients who had relapsed after transplant. If you relapse after transplant with a high-risk leukemia, your chances of survival are extremely low. Dan Egan is the PI of this ongoing protocol. Essentially what happened was we put 10 billion cells per meter squared at day 0 and 14. But for safety, the FDA had asked that no subsequent infusion were administered if there was a nice persistence of the T cells, more than 3% of the CD8 T cells. We screened a lot of patients for this. As you can imagine, patients going through transplant, a lot of them drop out, a lot of them have complications, but we were able to treat 12 patients in the prophylactic arm and 12 patients in the treatment arm. Infusing, we were, pleasantly surprised that the toxicities of these T cell infusions is very, very limited. Patients will have fevers and chills maybe for 24 hours, within 24 hours, usually lasting less than that. And then they just go on their way, despite the fact of having really nice persistence. If we look at our 12 patients on the prophylactic arm, now we have about 2 years of follow-up, and none of them have relapsed. We compared this to a matched comparative group, so patients who had undergone transplant at the same time at the Hutch, but who had not received T cells for multiple reasons, including the fact that they were not HLA matched with our A2 restricted TCR. And in these patients, they were selected not to have relapsed at day 28 to 35, which was the time that these patients became eligible to receive our T cells. And indeed, those have relapsed about 75 to 80%. And this is statistically significant. Here's just showing the matched risks, showing that the comparative group is similar to our patient population. So is our T cells responsible for that? So remember that these patients have no disease after transplant and they have not relapsed. So we cannot question whether their leukemia, we cannot have a sample of their leukemia to answer specific questions. So we have to go about it in indirect ways. So the first thing we looked at was T cell persistence. And indeed, there we saw that many patients had really very robust persistence, about 40, sometimes up to 50% of their whole CD8 T cell population now carried the transgene. Were these cells activated when we looked at PD-1, which is an activated exhaustion marker? Yes, they remained activated. And when we look at the exhaustion, so long-term exhaustion, one question you can ask looking just at the T cells is if the T cells will recognize its target, it will slowly change in phenotype, slowly become exhausted. And indeed, this is exactly what happens on these, but on a very, very long scale. We looked about up to 100 days, and it took about 100 days for the effector memory population to slowly decline, be replaced majorly by a terminally differentiated population. But also there was this central memory population also being formed, suggesting that now these cells were retaining a small population of cells which could potentially stem effector memories and central memories for tumor control. So this is all indirect ways to look at this issue. We also did single cell RNA sequencing and our CD8. So now we can identify our T cells, infused T cells, because they have a specific introduced gene, and these actually cluster in the CD8 effector population majoritarily. This is a patient who is about 100 days after his infusion, and also in the central memory population. They cluster together, meaning that they are pretty similar to normal CD8 T cells. But when we looked very specifically at differences, we saw that the transgenic T cells were slightly more activated than the endogenous T cells, secreting lymphotoxin beta, granulolysin, and granzyme K, which actually was also in another patient. So here, looking at this indirectly, we say, well, our T cells are there, they're staying. As an immunologist, I would say, you know, a T cell is there for a reason. And so, you know, we think in this prophylactic arm that these T cells, we have indirect ways of saying, yes, they were indeed responding, and they were not responding. I'm not showing this data for the sake of time, but they were not responding to the EBV. So what about our prophylactic, our treatment arm? And as I said before, we learn a lot from our failures. We did see evidence of direct anti-leukemic effect. Here is a patient who had bone marrow detectable disease, got the T cells eliminated. She's doing fine. However, when we looked at overall survival of all the 12 patients together, we did not really see any difference compared to the comparative group. So why? So we started to look at persistence, because we know that, you know, if there's going to be a response, we're going to have T cells around. And in this particular cohort, a lot of them had received CMV-specific cells. And those CMV-specific cells here in green really did very poorly. And we would try to put more cells in, but there was nothing we could do. The persistence was just not there. It turns out we did a whole where we had to go all the way back to the donor to look at the phenotype of these CMV-specific cells, and it turns out that CMV-specific cells compared to EBV are much more exhausted, meaning that they are effectors, whereas EBV-specific cells are more central memory. And we know from work by Sandra Dell that actually effectors don't do that well. So moving forward, obviously, we are going to be selecting EBV-specific cells. So that was one reason why it didn't work. But we still had 2 patients here that had, nice persistence, and both of them actually progressed. One died of his disease, and we'll look at his case in more detail. We looked at WT1 expression. One patient with persistent T cells had very little expression, so that was one of the reasons. We looked at class 1 downregulation, and that was not the reason in any of those patients. So let's look at this particular case. So here, this person here is a 27-year-old man with this WT1 positive AML. Here I'm showing his AML. It's screaming high for WT1, so he certainly has the antigen that we're targeting. And this patient had relapsed after his second allogeneic transplant, had received chemotherapy, got rid of the AML, and then received the T cells. And the T cells had persisted quite a long time, actually a whole year, before his AML relapsed. And we were we could take 2 samples for single cell RNA sequencing. So we took 1 sample at the time of response, and which did not contain any ML, and then 1 time point at the time of progression, and started to look and ask the question: Are his T cell activated? Are the T cells infused just sitting there doing nothing? Or are they actually involved in holding his disease? What I do want to say is that for a second relapse after after relapse of a second allogeneic transplant, patients don't usually stay without disease for a whole year. So that's very unusual. So here, let's look at his T cell. So here, of his, the single cell in his blood. So we're looking at all these populations, and these are 2 time point overlapped, and you can see that they cluster nicely. At the time of response, so now we're separating the time of response compared to relapse. The time of response, you can see he's got CD8 T cells, got CD4 T cells, got monocytes, and he's got the second population here in green of CD8 T cells, which is completely separate from these other CD8 T cells. You can also identify NKs and B cells. This population completely disappears at the time of relapse, and now you can see circulating AML in the blood. When we look at where our transgenic T cells are, it turns out that they completely overlap with this green CD8 T cell cluster. So basically the T cells are now clustering completely different. So they're very, very different. And you can see that in 3D. And when we look at what are the differences between the green and the green CD8 T cells, which contain the transgene, and the other ones, so the non-transgenic ones, they're very activated. There's a lot of genes suggestive of recent activation, including CD69, CTLA4, anti-interferon gamma. So here we have, at the time of response, this very activated T cell population. So let's try to figure out how probably responsible for tumor control. So the next question is, well, how did the tumor escape? In parallel, when we were looking at that, we started to look at protein processing and peptide presentation. Remember, cells have this mechanism where they have a proteasome where they cut up peptides that are then taken up by the HLA and presented on the surface. There's two types of proteasome. There's the standard proteasome and the immunoproteasome, which is more upregulated in terms of inflammation. And cells can go from one to the other. And what we do know, however, is that our specific epitope, the peptide that TCRC4 targets, is specifically processed by the immune proteasome. So you have to have those three subunits, beta 1I, beta 2, and beta 5, in order for TCRC4 to be processed. And this was actually very beautiful work from Nathalie Vigneron and Benoit van den Einde, showing that the specific proteasome subunit that you absolutely need is the beta 1I. So in parallel to this trial, we started to look at other epitopes, also HLA-A2 restricted of WT1. And here I'm showing, so we selected this particular cell line, which is only expressed the standard proteasome, because we wanted to find another epitope that would also be processed by the standard. And when you take this RKO cell line and you try to kill it with TCRC4, it does not. But when we looked at other epitopes, we did a whole screening, but we found another one that was now able to kill this cell line that exclusively expressed the standard proteasome, so it was not immunoproteasome dependent. And in our patient, when we looked at AML, we saw specifically that this beta 1I subunit of the proteasome was overexpressed in the relapsed AML. And so here we look now at the two TCRs. So here we're taking fresh leukemia. We're looking at both TCRs. So remember, they recognize WT1, but just little different parts of WT1. They both kill nicely fresh leukemias. And then when we take the leukemia of our relapsed patient, we can see that his immunoproteasome is completely lacking beta 1I. Beta 1 in the immune proteasome is completely lacking, whereas the subunits of the standard proteasome are nicely expressed. And indeed, when we take this patient's blood at the time of, so leukemia at the time of relapse, we can kill it using our new T cell receptor, which is processed by the standard proteasome and is much less killed by the T cells that were actually hanging around there, but had lost effect. So again, I think that even in a real world targeting a self-antigen, we are now able to say, yes, our T cells are there. If they hang around, they're probably there for a reason. So they certainly are having an effect, but they are so active in delaying relapse, but they're not potent enough to prevent acquired resistance, which I think the two cases that I've shown you are both one in the solid tumor world and one in the AML world of acquired resistance. So what are we doing moving forward? I'm getting up against the time, but I will describe a little bit what we think are ways to go about these problems. So first of all, target more than one specificity. I think that if we put too much pressure on one particular epitope, the tumor is so labile, it can now mutate. So if we, for example, targeted an HLA-A and an HLA-B epitope, maybe on the same protein, we could have more chances of success. we can look at targeting more than one protein. I haven't shown you data, but these AMLs express other protein that also can be targeted by T cells. And one other component of the immune system that we sort of forget, right? Because we say, okay, well, CD8 T cells are killer cells. Let's try to activate those. But CD4 T cells are really important also in the help that they provide to the CD8 T cells. And now we actually have ways in order to engage class 1 TCR, so a CD8 restricted TCR on the CD4 T cells, and we can do this by either having high affinity TCRs or co-transducing the CD8. So basically the complex can form on the surface. And then one other aspect of our TCRs that I was alluding to in the beginning is that they don't carry a signal to. This is the natural protein. And so what can we do now that we have ways to be able to manipulate genes? What can we do to try to put a signal to in the T cell receptor? It turns out you can't just stick it in anywhere. It doesn't work. So there are other ways that you can use the other components of the immunological synapse to plug in, so artificially plug in a signal to the intracellular portion of CD28 and 4-1BB. And those enhanced T cells, I think you will start to see them appearing in the clinic probably within the next 1 or 2 years. Okay, I'm going to stop here. I do want to acknowledge, so obviously this is work, it's very, very collaborative work. I started out in Phil Greenberg lab, and I continue to collaborate quite a bit with him. Paul Niem with the, and his group with the Merkel cell carcinoma. Castian Yi, I just figured out that he was also a Fialkov Award a long time ago. Now he's in MD Anderson. The clinicians, people in my lab, Kelly Paulson, Megan McAfee, which are just delightful to work with, and my funding for now. Okay, thank you very much.
We don't have time to carry on, so if you don't mind repeating questions.
Oh, okay, yeah.
We have time for questions. We've got 5 minutes for questions.
Very fascinating, some of the issues. I wonder if you can tell us why CMV versus EBV. CMV doesn't persist and the EBV does, and is that an option then for some kind of co-stimulation or co-immunization on an ongoing basis to keep those T cells persistent?
Yeah, okay, so the question is, why does CMV specific cells and not EBV don't persist and EBV do? So I think it's got to do with it's unclear what really is the mechanism, but I believe it's got to do with some kind of epigenetic regulation, meaning when we actually looked at those products that we infused between the CMV and the EBV, there was absolutely no different. And we went back and, we said, okay, well, maybe is it at early stimulation? We looked, no difference. We had to go all the way back to the original donor cell. So the donor had given the leukapheresis, and we looked at the EBV and CMV specific cells in the donor, and that's when we started to see real differences, where EBV specific cells are they express very much a phenotype what we call central memory, so CD62L, CCR7, so they're long-lived cells. Whereas CMV-specific one had more of an effector memory, low CD28, CD127, absolutely no CD62L or CCR7, suggesting that they had been recently activated. And Stan Riddell actually did work in monkeys, showing that if he he took central memory cells and effector memory cells, the effector memories would not persist. It was only the ones that were derived from the central memory cells that persist. And I think this is exactly what we're seeing here. Turns out that the biology also of CMV and EBV is different. CMV in normal donors is reactivated quite often, whereas EBV, you really have long-lived memory cells. what I am curious is whether, EBV in normal donors, it's all healthy donors, it's also sometimes reactivated. But what about, T cell responses, for example, to a vaccine where, a person sees it once and then creates a long-lived memory, and might those cells actually be better after transfer? We are, just to follow up, we are looking into now, in the autologous setting, we don't really have to select the endogenous T cell receptor. We're not restricted that much. And so we are looking into central memories subtype, so versus naive subtypes, in order to see if after T cell infusions we see better. But it could be that, EBV specific cells or central coming from a viral population might be actually quite successful as substrate cell after transfer. There's not a lot of them, unfortunately. Okay, thank you very much.